Now on to the end of macromolecules. We've got two left. The first is going to be nucleic acids. Now this one's kind of fun because the major guys that contain nucleic acids have it in their name. So the two common guys that we'll talk about is going to be DNA and RNA. These are the two big examples that we'll discuss. And they stand for deoxyribonucleic acid and ribonucleic acid. So they both tell you pretty much what they are with their name. So just remember the Na part stands for nucleic acid. Now nucleic acids, which we'll talk about a lot more in depth later when we get into genetics, these guys are going to essentially store the information that helps determine who you are, like how to build you. And they're also going to transfer that information, or at least about half of it, via sperm or egg to create an offspring that's going to contain that information so that your offspring is able to grow and develop as well. So you're going to be able to use this nucleic acid to take the information that's stored in you and essentially pass it on to your offspring to the next generation. So this is incredibly powerful stuff that allows for populations to grow, populations to evolve and adapt. It's critical that we have this particular molecule. Without it, we could not develop to be us, and we certainly could not develop to produce offspring that are like us, and in some cases slightly different from us, which gives us the ability to adapt as time goes on. So critical molecule. Now it's composed of a monomer. In this case, the monomer is going to be a nucleotide. Now a nucleotide is composed of 3 parts. So this right here is going to be 4 nucleotides. I'll kind of delineate them for you. So each nucleotide is going to have a 5-carbon sugar. So this is going to be our 5-carbon sugar, and that's the guy in the middle here. And that guy's in between a phosphate group, which is what that P is. All right, so we've got phosphate. And then at the top here, we have something called a nitrogen base. That's the big deal. You'll see the nitrogen base is the one that's typically A, C, T, or G, if you've heard of those letters. We'll get way more into them later, but it's the particular sequences of those things you have, of those A, T, C, G, and U if you're talking about RNA. It uses U instead of T. But the order of those molecules over hundreds and thousands of these nucleotides is ultimately what determines your genes, which determines what proteins you have, which determines who you are. So it's incredibly powerful which of these you get. But each nucleotide is going to be these 3 pieces, and then they will connect together, so you get these long chains that can consist of millions of these nucleotides to make up one piece of DNA or one piece of RNA. The easy way to distinguish between RNA and DNA is DNA always has a second strand. This one does not. You'll see it's just one string of nucleotides. So this one is ultimately RNA. You'll also see it contains a U. Whenever you see U, it's going to be RNA. If you see a T, it's going to be DNA. And DNA, as I said, would always have another strand that's next to this. Like the A would be attached to a T here. You know, the G would be attached to a C, and then you'd have sugars and phosphates on the other side as well. So we'll get more of that later. And these guys are going to be composed of a few different elements. When we talked about the first 2 guys, they were primarily carbon, hydrogen, and oxygen. When we talk about nucleic acids, they're also going to have some nitrogen, hence we call it a nitrogen base. You can see the nitrogens. And they also have a phosphorus. And you can see that because they have a phosphate group. And so if we're looking at a particular molecule, and we know that it's a macromolecule, we can oftentimes analyze it and say, does this contain nitrogen and phosphorus? If it does, there's a good chance that what we're looking at is ultimately a nucleic acid. So it can be used to help identify stuff. Now proteins, these are possibly the most important guy that we have in our body. You could argue it's nucleic acids because nucleic acids code for the proteins that we have. The nucleic acids are kind of like the blueprint, and the proteins are like the actual construction workers that get stuff done. So proteins pretty much make us who we are. Any pigment we have, that's a protein. The enzymes we have, proteins. Many of the structures that we have and the things that allow stuff to transport in our body, proteins. You know, in our blood, what holds oxygen and transport it, that's a protein. So pretty much what makes you you is going to be proteins. If you're unsure about something and you need to guess a macromolecule, usually a good choice to guess protein, all right? So they're made up of a bunch of these particular monomers called amino acids. There's about 20 different types of amino acids that we possess within animals that we can stick together in all kinds of different combinations to give us proteins. So this should give you a sense of just how many proteins that we could make. Because if you look at a particular protein that might consist of hundreds or thousands of amino acids, and for each particular spot, from the first spot to the 1,000th spot, we have a choice of 20 different amino acids that we could put there. And if we change even a single amino acid, we will get a different protein that often has a different function. You start to realize there's a ton of possible proteins out there. A ton. And now this will be made of, once again, about 5 different elements. We've got carbon, hydrogen, and oxygen. These are our standbys. Pretty much all organic stuff tends to have those 3 guys. Then we've got nitrogen, which we also saw in nucleic acids. The one that kind of indicates protein here is they tend to have a fair amount of sulfur. So if you see sulfur, that one is the big indicator that, okay, this is a protein. That's my red flag. Much like we took a bunch of monosaccharide monomers and got a polysaccharide, if we take a bunch of amino acids, we're going to call a whole bunch of those bonded together a polypeptide. Now, for our purposes, we're going to consider a polypeptide about the same thing as proteins, but in reality, you'll see that some proteins consist of more than one polypeptide, so they can actually be constructed by taking a series of these long chains of amino acids, multiples of them, and sticking those together to make one final protein. So it's kind of like if you had the transformer bot that was like 4 different transformers could fit together to make one bigger transformer. That is ultimately what you'd have with a protein. So a protein is either one or more polypeptides if you want to get technical, but for most of the conversations that we have, I will let you use them pretty much interchangeably, but I want you to realize they're not identical. And we've talked about this with enzymes, so I'm going to bring it up again, because enzymes are proteins. Proteins are going to fold up, so they've got this long chain of amino acids, but then that long chain of amino acids starts to kind of see other particular amino acids that are a little ways away from it on the chain, and that guy might be polar or that guy might be non-polar, and so they find guys that they like, that they're attracted to, and then they tend to bend, and then they might hydrogen bond with them, or they might just like to stick near each other because they both hate water, and so they'll start to fold up. Now, it's critical we know how proteins are folded because the order of the amino acids doesn't tell us the whole story. We have to figure out which parts of the protein like the other parts and are the right distance away that they can fold up and interact, because that's ultimately how we have to calculate what the final shape is of the protein, and that final shape, that final 3D structure determines its function. And if we change the shape, this is what we talked about when we used that word denature in the enzyme lab. If we heat something up or expose it to pHs that are not appropriate, it can make it shift a lot of those things. It can make it shift the folds. And that will change the shape, which can change the function of that particular protein or that particular enzyme. Remember, enzymes are just a type of protein. All enzymes are proteins, not all proteins are enzymes. All right? So because of that, you'll see the shape of a protein will be critical when we discuss it. Anything that you do, even if you don't actually change, remove, add amino acids, if you change the way the amino acids fold, you are going to change the function of that protein, and so it may no longer work properly.